Hello, everyone, and thank you to the conference organizer and to all for joining today. My name is Negin Taghehchian. It's an honor to speak with you about a topic that bridges traditional Persian medicine and modern system biology. That names Saffron and gut microbiome, possibilities for glioblastoma treatment and prevention. In this slide, I want to introduce my colleague that participates in this study, Dr. Aryoshahin Jafari as a corresponding author, me and Dr. Aida Abbasi and Hakime Karami. In this slide, first I want to introduce the glioblastoma. Glioblastoma is the most common lethal brain tumor in adults. It's grade 4 astrocytoma with a rapid growth and strong invasiveness. Even with surgery, radiotherapy and temozolomide survival is usually under 15 months. The tumor heterogeneity, diverse cells and pathways, resistant stem cells, and its complex microenvironment, including macrophage angiogenesis and hypoxia, make treatment extremely difficult. The main challenges included the blood-brain barrier because of the limitation of the effect drug delivery and persister cell cause recurrence in nearly all patients. And also highly invasiveness of this tumor make it impossible to complete resection of this tumor. Here I want to tell about the key pathophysiological mechanism that plays critical roles in glioblastoma. As you see, loss of PTEN activates PI3-AKT mTOR pathway and EGFR amplification, which promotes cell growth and survival. Loss of TP53, which disrupts cell cycle control. TERT promoter mutations, which maintain telomere length. MGMT promoter methylation, which enhances responses to temozolomide, PI3-AKT mTOR, and MAPK-ERK signaling, which drives proliferation, invasion, and therapy resistance. And also hypoxia, which induce HIF1 and VEGF upregulation and leading a strong angiogenesis. And also overexpression of PD1, PDL1, and CTLA4 upregulation, which causes immune invasion. And also beyond these classical pathways, non-coding RNAs that included long non-coding RNA, microRNA, and circular RNAs, which add a new layer of gene regulation and control tumor behavior in GBM. And also dysregulated non-coding RNAs like H19, PBT1, hot air, and also other long non-coding RNA participated in angiogenesis, apoptosis, migration, and also proliferation. Sorry, just to confirm, are you changing my slide? Are you sure that's my slides that changes with myself? Sorry, did you hear my voice?
Yes, Doc. Hello.
Yeah.
Yeah.
Yeah. Thank you.
You wanna change the slide?
No. I want to confirm that. Did you see my slides and hear my voice?
Yeah. It's perfect.
Okay, thank you.
You can carry on.
Okay, I want to tell you about the factor in GBM that play roles in GBM. As you know, genetic and environmental convergence shapes tumor heterogeneity, immune response, and also therapy. Exposure to various environmental pollutants, including endocrine disruptors, airborne toxin, pesticides, heavy metal, and water contamination, increase the risk of brain cancer. These agents can cause DNA damage, oxidative stress, hormonal and signal disruption, and immune imbalance, which is leading to brain tumor initiation and progression. Additionally, ionizing radiation and an electromagnetic wave acts as a physiological carcinogenesis. As you see in this slide, ionizing radiation and short wavelength electromagnetic wave have an important role in contributing in glioma and glioblastoma development. By contrast, the influence of long wavelengths, such as those used in audio and visual communication technologies, remain less clear. Before 2018, many studies suggested potential biological effects. For example, in this article that published in 2009, publication that is reported the direct association with these brain tumors. Additionally, recent research by Dr. Shahin Jafari demonstrated effects on Candida albicans, a fungal species that exists in our environment and interact closely with the human body that shows here. As research advanced and likely as communication devices improved in safety finding became more inconsistent, some studies have even explored possible therapeutic application of these long wavelengths waves. According to Dr. Shahin Jafari's more recent perspective, the primary concern may not be the electromagnetic waves themselves, but rather the noise associated with them. A related patent has been registered, although explaining the full concepts of this article is not possible because of the time of this presentation. As you know, all of us faced with the glioblastoma, that is one of the most aggressive and deadly brain tumor. Even today, despite major scientific and medical advances, we still struggle to extend patient survival beyond 3 years. As you know, after more than 12 years experience in cancer research, this work has become more personal to me. I am collaborating with Dr. Shahin Jafari, an expert in glioblastoma, microbiology, and immunology. Together, we are developing a new saffron-inspired approach to reflect both my culture and my personal roots in saffron-producing region, as you know, Khorasan, Iran, aiming to bring a new hope to better outcome for people with glioblastoma cancer. Now I want to introduce you about why gut microbiome. The gut microbiome plays central role in regulating systematic inflammation, metabolism, and immune balance. It communicates bidirectionally with brain and other organs through neural, immune, and metabolic pathways. Microbial metabolites such as short-chain fatty acid and tryptophan derivatives can cross the blood-brain barrier directly, influencing brain function and inflammatory tone as well. When this balance is disrupted, a condition called dysbiosis, it causes neuroinflammation, immune suppression, and even glioma progression. Importantly, certain bacterial toxa, including Akkermansia, Lactobacillus, and bactericide, have emerged a potential biomarker that may predict response to radiotherapy and immunotherapy. Altogether, understanding and targeting gut microbiome could open new therapeutic strategy for GBM treatment as well. In this slide, I want to tell you about the gut-brain tumor axis in glioblastoma patients. Recent studies have revealed a strong connection between the gut microbiome and glioblastoma progression, which is known as gut-brain tumor axis. Gut dysbiosis also affects the GBM drivers such as EGFR, PTEN, and TP53. The gut biopsies also play with patients have reduced gut microbial diversity, increased inflammatory bacteria, and higher interleukin 6 and TNF alpha, which promotes neuroinflammation and tumor growth. Microbial metabolites can cross the blood-brain barrier and reshape microglia and T-cell responses, which contributing to immune suppression and thereby resistance. Targeting the microbiome, including which bioactive compound like saffron, may shift GBM toward a more immune active and treatment responsive states as well. Now I want to introduce you the saffron, the gold healer of mysterious Iran Persia. Saffron is a medicinal spice rich in several bioactive compounds, mainly crocin, crocetin, and safranal, which are responsible for its color, taste, and aroma, as well as its therapeutic potential. As you know, crocin is a water-soluble carotenoid glycoside. It exhibits strong antioxidants and anti-inflammatory properties by inhibiting NF-kappa B and MAPK pathway and show anti-tumor effect in various cancers. Crocetin, a decarboxylic carotenoic acid formed when crocin is broken down, is a lipid-soluble and can easily cross a biological membrane. It also induces apoptosis, modulates oxidative stress, and enhances oxygen diffusion in tissues. Safranal is an aromatic aldehyde responsible for saffron's characteristic aroma. It helps stabilize immune tone and has neuroprotective and anti-anxiety effects as well. Altogether, saffron represents a remarkable example of how traditional Persian heritage and modern biomedical science intersect. And also, as you see in this picture, saffron plays a role in antioxidant, anti-cancer, anti-anxiety, anti-Parkinson, anti-inflammatory, anti-Alzheimer, and anti-diabetic, and anti-depression. Uh, in this slide, I want to tell you about molecular mechanism of crocin against GBM. As you show in this slide, I will tell you about this article that shows the crocin anti-tumor effect in glioblastoma. It induces apoptosis through caspase-3 activation and BCL2 downregulation, and induce differentiation by increasing beta-3 tubulin and NFH markers. And also it inhibits PI3 AKT and ERK MAPK pathway that supports tumor survival. It also reduces ROS and angiogenesis and also enhances temozolomide sensitivity in GBM cells. And also they do animal studies and shows reduced tumor volume and improved survival as well. Overall, it acts on multiple pathways to suppress tumor growth and boost treatment response in GBM patients. Next, I want to tell you about molecular mechanism of crocin against GBM. As you see in this slide, first of all, in this study, they shows that combination of caffeic acid phenyl ester as the small form is safe, derived from B. polypylus, and crocin shows a strong synergetic effect against glioma cells, which is leading to a significant inhibition of glioma progression. As you see in this picture, when you combine CAPE and crocin with each other and treat glioma cells, it decreases proliferation, colonogenicity, and also the expression of EGFR, and also increase the cell death, apoptosis, and expression of p53 and pcDNA. In another study, it shows that crocin induces apoptosis through p53-dependent pathway in C6 glioma cells, which is leading to a strong growth inhibition, and this suggested that crocin may be a promising therapeutic agent for treating drug-resistant gliomas. As you see in the other article, the cytotoxic effect of crocin on glioma cells, it shows that crocin shows inhibitory effect on glioma cell growth, which is reducing cell viability and proliferation in dose and time-dependent manner. Its higher concentration and long exposure significantly strengthens this anti-proliferative action, suggesting crocin's potential as promising natural anti-cancer compound. And the other study it's an in vitro study of saffron carotenoid in different cancer cell line of glioma. It shows that crocin treatment significantly reduced glioma cell viability and proliferation in dose and time dependent manner, accompanied by increased ROS production and activation of anti-apoptotic pathways such as caspase-3. It also downregulated anti-apoptotic protein BCL2 and inhibited PI3 AKT and ERK MAPK signaling, which are both crucial for tumor survival. In conclusion, crocin can suppress glioblastoma growth by inducing oxidative stress and apoptosis while blocking the survival pathways. In the next slide, I want to tell you about a molecular mechanism of safranal against GBM. This study investigated the anti-tumor mechanism of safranal by combining network pharmacology, molecular docking, and in vitro validation. it shows that safranal can interact with PI3, AKT, mTOR, and MAPK and p53 signaling pathways, which is critical regulator of cell proliferation and apoptosis in GBM. And also docking simulations shows a strong binding affinities between safranal and core GBM related proteins such as AKT1, TP53, and also EGFR as well, which suggested that safranal can modulate tumor growth and survival. In experimental design, safranal significantly reduce viability and migration, and also inhibit proliferation and invasion, and also promote the G2M phase arrest and apoptosis, and also inhibit the PI3 AKT mTOR axis and cooperate with TZM to inhibit cell growth. In the next slide, I want to tell you about the trans-sodium crocinate, that is a small synthetic molecule derived from crocetin. I want to tell you about the molecular mechanism of this molecule against GBM, which is act as oxygen diffusion enhancer, which improving oxygen transport through plasma and the tissues. Tumor hypoxia is a major cause of resistance to therapy in glioblastoma. As you see here, this study has a clinical trial registration number as mentioned here. And this study shows that by reducing hypoxia, TSC increased the sensitivity of glioblastoma cells to radiotherapy and chemotherapy. This study combining TSC with temozolomide and radiation, which shows improved patient survival and good safety, that highlighting TSC as a promising radiosensitizer for high-grade brain tumors. So I want to tell you about saffron effect on gut microbiome in two separate articles. As you see in this article, saffron compound, especially crocin and crocetin, modulate gut microbiome balance by increasing beneficial bacteria and reducing harmful one. They demonstrate inflammatory genes such as NF-kappa B, TNF-alpha, and interleukin-6 and LPS that they're reducing inflammation and also improving gut health and potentially preventing colorectal cancer. However, high doses or prolonged exposure to crocin may induce opposite pro-inflammatory effect, which emphasizing the importance of balanced dosing. Gut microbiome plays crucial role in bioactivation of saffron compounds. And in the other studies, it shows that the crocin are converted by intestinal bacteria into crocetin, the active and absorbable form of this saffron. Without this microbial conversion, this absorption and biological activity are minimal. Therefore, individual differences in gut microbiome can significantly influence the antioxidant and anti-inflammatory effects of saffron. Saffron compounds also increases short-chain fatty acid production and mucosal integrity and balance the T helper 17 and T regulatory axis to maintain immune homeostasis. So I want to tell you about a molecular and microbial crosstalk in GBM. This crosstalk illustrates the complex interactions between saffron compounds and GBM pathways. This multi-target mapping shows over than 40 genes that are participated in this interaction, which highlighting the significant impact of saffron. Saffron compounds also increasing the microbial bacterial like Lactobacillus bifidobacterium and increases Clostridium that while reducing the Clostridium one. Also affect immune mediator like interleukin-6 and interleukin-10 and TNF-alpha, which plays crucial roles in inflammation and immune regulation. Saffron also influences brain-related pathways, including NF-kappa B, PI3, AKT, which involves in cell survival, inflammation, and cancer progression. Saffron also enhances short-chain fatty acid and butyrate levels of expression, which have anti-inflammatory properties and support gut health. These visual networks reveal that saffron compounds impact microbial communities immune responses, and brain pathway in glioblastoma as well. So in this slide, I want to integrate in traditional and modern medicine. As you know that AI-driven personalized microbiome and saffron therapy. As you know that AI is nowadays trend for using and knowing about the personalized medicine and personalize these drugs for each person with each criteria. As you know, AI models predict genomic, metabolomics, and microbial pattern, which is links to GBM risk. Adding bioinformatics with metabolomics change the way of treatment and personalized saffron dosing and enable personalized therapeutic pathways as well. This goes to prevention in at-risk individual, family, and genetic exposure family with genetic exposure of this kinds of cancers, and we can medicate them in the best way. So we designed a clinical trial design for GBM with our hypothesis. In our clinical trial design for GBM, we will start with pharmacokinetics and safety studies in human to ensure safety and proper dosing. We will also evaluate blood-brain barrier penetration and how well the drug reaches to brain. Next, we plan to combine it with standard therapy like surgery, radiation, and temozolomide to test for synergy. Our phase first and second design will focus on dose escalation, safety, and early sign of efficacy. We will integrate key biomarkers like CT-Chelachar and neuronal markers that are related to GBM, and finally, we are standardized saffron-derived compounds extraction and purity to know and ensure the consistency. And now is the future of prevention, and we want to know how to prevent these kinds of cancers like glioblastoma with our hypothesis. We combine framework like identify microbial biomarker for GBM risk and map saffron microbiome synergy via metabolomics, and also validate multi-omics predict model in cohort study to build predictive model for GBM risk or treatment response in real patients group as a cohort study. And also collaboration across bioinformatics to know about the data analysis and modeling of this cancer, and also using neuroscientists for know better about the brain biology and GBM patients, and also ethnopharmacology, which is the study of traditional medicine like the saffron, which aims to reprogram gut-brain tumor microenvironment. And the ultimate goal is to change the communication between the gut, the brain, and the tumor, which is using natural compounds like saffron and microbiome modulation to make the body's environment less favorable for tumor growth. So thank you for your attention. And if you have any question, this is our contact information of Dr. Aryo Shahin Jafari and his LinkedIn link, and it's mine to have. If you have any question, feel free to ask.